Let's talk about DNA replication. This process is done in order to pass on identical DNA to daughter cells. Okay, so this process is DNA being copied to DNA. So you have one DNA becomes 2x DNA. Okay, so for example, it is used for mitosis, which is a form of cell division that creates identical daughter cells. So here, in this process, you may have, say, one cell here, and it has one complement of DNA of chromosomes. After replication, that same cell will now have two copies of the same chromosome. So this is what DNA replication does. And then when this cell undergoes mitosis to form two daughter cells, the DNA will be split, one going to each daughter cell. And so the daughter cells now have the same identical cell as the original cell. And so you only do DNA replication prior to doing cell division. Okay, so this happens during the it's called the S phase. Occurs during the S phase of interphase. That's when you double your chromosomes or double your DNA. So let's look at how this works. Okay, so if you had a double-stranded DNA, here's one strand. Here's your other strand, okay? Open them up, and you can do this as well if you want to, like that. All right, so the DNA is double-stranded and helical shaped. So you open it up like this to expose two strands, and then once opened up, you have an enzyme that goes from this end. This is a DNA polymerase enzyme. DNA polymerase enzyme will go from this end and copy DNA that way. And one will go from this end and copy DNA this way. Okay? Now, the enzyme that unzips it here, the unwinding enzyme is called DNA helicase. That's the enzyme that will unzip your DNA and allow for then your DNA polymerase to hop onto it and to make the strand in two directions. So one strand will go this way. So here you'll make DNA going that way. And one strand will be made going this way. Okay? And so imagine that then as the helicase unzips more DNA, right, then this one that's already going this direction will continue to make more, like so, continuously, while this one is already down here, so then this one will have a new polymerase start back here. Well, it's how it is. So if you have two strands like this, two strands of DNA, one strand that's going towards what's called the fork, the replication fork, will be made like this continuously, and the other strand will be made in pieces. So each time you expose more DNA, that strand will need a new start to come this way because it's going away from where you have new material. Okay, so the strand is made towards the fork. This opening here, by the way, is called your replication fork. So the strand that's made towards the fork is called your leading strand. And the strand that's made away from the fork is called your lagging strand. Not sure if these terms are really used anymore, but lead strand going towards the fork, lagging strand away from the fork. And so when you make it in these jagged pieces are also given a name. They're called your Okazaki fragments. Okay. And these Okazaki fragments are ligated eventually. They're joined together by an enzyme called DNA ligase. So this enzyme will make them come together. So DNA ligase is the enzyme that will then join these two together. And at the end of the process, you'll have the old DNA strand up here, right? So if you can imagine that once you make a new DNA molecule in both strands, they will then fold with the old strand that was previously there. So basically, this becomes like this. And this strand is now folding with that. Okay, and same here. The old strand and the new strand. So now the new double-stranded DNA that forms will have an old strand and a new strand. So it's called semi-conservative replication. Meaning the new DNA you form will have an old strand and a new strand. Old strand, new strand. Okay? That's how we kind of preserve our heredity, passing down with more consistency, because we're not really making things brand new, we're just copying what's there already. All right, so that's your semi-conservative replication. So let's summarize a little bit here. The steps of DNA replication are: one occurs in nucleus only. DNA does not leave the nucleus, except for mitosis. And here's the steps are: DNA helicase unzips DNA. Step 2, DNA polymerase enzyme copies each strand of DNA. Copy it. And the DNA polymerase, same as RNA polymerase, by the way, DNA polymerase works in a 5 to 3' direction. Has to. That's why both of them can go the same way, because imagine that if this was your, say, 3', 5' strand, and this one would have to be your 3', 5' strand. And then when the DNA polymerase is making its new strand, it has to be made in the 5 to 3 direction, like that. So here is 5 to 3, 5 to 3. That's why it's going this way, because it's forced to go in this direction only. Okay. And 4, the strand. So we can name these two strands as leading strand is made towards the fork. Made towards the fork right here, towards your replication fork. And so that's called 4A. And 4B, lagging strand, is made away from the fork. Okay? The ligase strand, furthermore, is made in what's called Okazaki fragments, which are then joined by an enzyme called DNA ligase. Okay? And finally, at the end, old strand pairs with new strand. I was interrupted again. So old strand is paired with new strand, such as this. So you have old and new, old and new, and that's called your semi-conservative replication. Okay. All right. Of course, the polymerases, their job is to go in. For example, if the blue one, just so you're clear on this part. This is DNA, the old strand, let's say here, GCCCCCA. It's going to base pair with CGGGGGT, base pairs. And then when you make your new strand with the polymerase, it comes in and makes CGGGGT, which happens to be the same as this right here. And then the other polymerase will make GCCCCCA, which is the same as this strand. So as you can see, the strand that you're making is the same as the other old strand. That's why they can base pair together. So then these two will join together and these two will join together to form the new DNA molecules. Okay, that's replication. What else should I tell you about replication here? So it can be done in about 8 hours for a cell to copy all 6 billion DNA nucleotides, okay? You can do, the enzymes can do about 100 bases per second, and many polymerases will simultaneously make pieces of the DNA molecule. It's not just one DNA polymerase taking on the job of trying to copy your entire chromosomes complement. That's way too much work for it. So yeah, many thousands will get involved in this process. Okay, we stop there.